Welcome to the Cytokine Signaling Forum series of highlights from ACR 2021, where authors take us through their posters and presentations on cytokine signaling and JAK inhibitors. My name is Len Calabrese, and I'm the head of the Cleveland Clinic section of clinical immunology. In this podcast, we take a look at the ACR presentations that covered the basic science behind cytokine signaling of many rheumatic diseases, as well as some other clinical presentations I think that you'll find of interest.
So hello, this is Dr. Kato from Department Rheumatology, Endocrinology, and Nephrology, the Faculty of Medicine and Graduate School of Medicine, Hokkaido University. So thank you very much for giving an opportunity to present our recent work entitled JAK1 Regulates Autophagy and Reinforces the Inflammatory and Autoimmune Potentials in Rheumatoid Arthritis, synovial fibroblasts. Rheumatoid arthritis is pathologically characterized by autoimmunity against styrulinated proteins, proliferative synovitis, and ultimately joint dysfunction. Synovial fibroblasts are, when activated, capable of hyperproliferating and producing large amounts of pro-inflammatory mediators, including IL-6, thus considered to be the key effector cells in rheumatoid arthritis pathogenesis. We previously showed that synovial fibroblasts obtained from RA patients have high autophagy activity, probably contributing their active phenotype. Besides, our recent study suggests synovial fibroblasts' autoimmune potential by demonstrating the increase of styrulinated vimentin and its interaction with MSC class II when treated with interferon gamma and autophagy inducers. JAK1 is an emerging therapeutic target in RA, but its roles in the active phenotype of synovial fibroblasts remain to be elucidated. Given this background, this study aimed to clarify the role of JAK1 and the regulation of autophagy and in the inflammatory and autoimmune potentials in synovial fibroblasts. So this study was performed by following procedures. Synovial fibroblasts were derived from synovial tissue specimens obtained from RA patients during joint replacement surgery and used between passages 4 to 8 for all experiments. To inhibit JAK1, synovial fibroblasts were treated with its selective inhibitor, upadacitinib, for 6 to 24 hours with the optimal concentrations determined by BRD assay. To induce autophagy, synovial fibroblasts were starved using serum-free medium for 2 hours or treated with 10 micromolar of the proteasome inhibitor MG132 for 24 hours in the presence or absence of 5 millimolar of the autophagy inhibitor 3-methyladenine. The expression of autophagy-related genes, including BACLIN1, ATG5, and ATG7, were analyzed by real-time PCR. The expression of IL-6 was measured in cell culture supernatant by ELISA. The interaction between styrulinated vimentin and MSC class II were analyzed by in situ proximity ligation assays. So now I would like to move on to the results. So please look at figure 1. The inhibition of JAK1 with upadacitinib resulted in the significantly decreased expression of autophagy-related genes, including BACLIN1, ATG5, and ATG7 in starved synovial fibroblasts. Similar results were obtained in synovial fibroblasts treated with other stimuli, such as starvation and interferon gamma. interferon gamma alone and interferon gamma with MG132. So next, please look at figure 2 regarding IL-6 production of synovial fibroblasts. The treatment of synovial fibroblasts with starvation and interferon gamma increased the expression of IL-6 in cell culture supernatants, which was inhibited by upadacitinib. Similar results were obtained in synovial fibroblasts treated with other stimuli such as interferon gamma alone and interferon gamma with MG132. I would like to move on to figure 3 regarding autoimmune potential of synovial fibroblasts. So red particle represents the interaction between styrulinated pimentin and MSC class II. Styrulinated pimentin MSC class II interaction was increased in synovial fibroblasts following treatment with starvation and interferon gamma. So the effect was canceled by the addition of upadacitinib or the autophagy inhibitor 3-methyladenine. The current results indicate that JAK1 is involved not only in cytokine-mediated signaling, but also in autophagy-inducing stimulus, such as starvation. Therefore, the inhibition of JAK1 may strongly suppress the active phenotype of synovial fibroblasts through these two pathways. Further investigation is currently underway, such as the effect of JAK1 inhibition on sterolination and the interaction between synovial fibroblasts and CD4 positive T cells, which may confirm our results. We would like to conclude that JAK1 positively regulates autophagy and reinforces the inflammatory and autoimmune potentials in synovial fibroblasts. The mode of action of JAK inhibitors would include the mitigation of synovial fibroblasts active phenotype. That's all. Thank you very much for your attention.
Hello there, I'm Vibeke Strand. I'm an adjunct clinical professor in the Division of Immunology and Rheumatology at Stanford. And I'm going to talk to you right now about poster 0232, the impact of Cravacity, which is a TYK2 inhibitor, on patient-reported outcomes and health-related quality of life, a phase 2 trial in patients with active psoriatic arthritis. So this was an analysis that evaluated the patient-reported outcomes, which included patient global assessment of disease activity, pain, the HAQ disability index, fatigue, and health-related quality of life by the generic SF36, short form 36. And patients were randomized equally to receive either Cravacity 6 milligrams or 12 milligrams versus placebo, 66 to 70 patients per treatment group, and the primary endpoint was week 16. The demographic and baseline disease characteristics were similar across the treatment groups and are presented in table 1 of the poster. At week 16, which is figure 2, we see the mean improvements from baseline in both SF36 physical and mental component summary scores, the PCS and MCS, were significant and similar with both doses of Cravacity versus placebo. We then looked at the domains, SF36 domains in figure 3. And what we can see there is that the improvements with both doses were significant in 6 of 8 domains. So the essentially physical function, role physical, bodily pain, which are the physical domains that tend to be most affected by psoriatic arthritis, but also vitality, social functioning, and role emotional. The mean changes across these domains range from 7 to as much as 19.7, so they well exceeded the minimum clinically important difference of 5 across the domains. We then looked at the percentage of patients who reported improvements that met or exceeded MCID in these PROs, and that is shown in figures 4 and 5. And so what we see is with both doses, we get MCID improvements that are statistically significant in patient global and HAQ. And then with the 12 milligram higher dose, it includes 3 domains: role, physical function, and social functioning. Altogether, these result in numbers needed to treat of about 4 to 6.4, and we consider less than or equal to 10 to be economically and clinically meaningful. We then move on to figure 6, the proportions of patients who report scores that meet or exceed normative values in HAC, FACIT-F, SF36, PCS, and the physical domains. And these ranged from about 0 to 10% to as high as 10 to 31%. So 0 to 10% at baseline, up to 10 to 31% at week 16. And this is compared to 5 to 12% with placebo. Similar improvements were seen in the mental domains and the MCS scores, although traditionally and clinically, we know that there are higher scores at baseline. So this illustrates that patients with active psoriatic arthritis, even who have failed conventional synthetic and or biologic DMARDs, are able to report scores that are consistent with healthy individuals that don't have arthritis. And by the way, there's poster 750 that will show similar meaningful improvements in this population using the PSAID, disease-specific instrument. In conclusion, these data demonstrate that statistically significant as well as clinically meaningful improvements in patient-reported outcomes, including fatigue and health-related quality of life, were reported by patients with active psoriatic arthritis who received deucravacitinib treatment for 16 weeks. Thank you.
I am Anjaneya Chimalakonda from Bristol Myers Squibb, and today I will discuss the poster titled Selective Inhibition of Tyrosine Kinase 2 with deucravacitinib compared with Janus kinase inhibitors. First of all, I would like to thank the organizers for inviting me to present this talk. So as a background, tyrosine kinase 2 or TYK2 is an intracellular kinase involved in the pathogenesis of immune-mediated inflammatory diseases and regulates signaling and functional responses downstream of the interleukin 12, interleukin 23, and type 1 interferon receptors. Deucravacitinib is an oral selective allosteric TYK2 inhibitor with a unique mode of binding to the less well-conserved pseudokinase domain rather than the conserved active site in the catalytic domain. This unique mode of binding provides high functional selectivity for TYK2 versus other tyrosine kinases in various cellular and whole blood assays. This unique mode of binding also may provide robust efficacy and a differentiated safety profile due to decreased toxicity relative to other kinase inhibitors. Now, in terms of clinical experience with deucravacitinib, in a 1-year double-blind phase 2 trial in subjects with psoriatic arthritis, deucravacitinib was efficacious and well-tolerated versus placebo. Deucravacitinib has also been shown to be efficacious in phase 2 and phase 3 trials in moderate to severe plaque psoriasis, and importantly, had a favorable safety profile without clinical or laboratory parameters suggestive of JAK1, 2, 3 inhibition being observed. Deucravacitinib is also being evaluated in other autoimmune disorders like inflammatory bowel disease, lupus, among others. Now, coming back to this poster, the main objective of this analysis was to compare the selectivity of deucravacitinib versus approved Janus kinase 1, 2, 3 inhibitors like tofacitinib, baricitinib, or upadacitinib at clinically relevant doses and exposures. The first step in this analysis involved development of whole blood assays that measure activity of TYK2, JAK1/3, and JAK2 pathways. The basic format of the assay, including the stimulant and endpoint used, is shown in the table below. Once the assays were established, half maximal inhibition concentrations, or more commonly called IC50 values, of the TYK2 inhibitor deucravacitinib and JAK inhibitors like tofacitinib, upadacitinib, and baricitinib were determined using these whole blood assays. Since these assays were conducted in whole blood, no further correction of these IC50 values using parameters like plasma protein binding, for example, were considered. This schematic provides an overview of the subsequent steps in this analysis. First, pharmacokinetic profiles or plasma concentration time profiles of cravacity, tofacitinib, upadacitinib, and baricitinib were simulated using parameters derived from published population PK models or from internal reports, for example, for cravacity. The doses selected for these agents include both the doses that are approved for some of the JAK inhibitors and also ones that were evaluated in phase 2 or 3 trials. The PK profiles were subsequently integrated to compare the plasma drug concentration in relation to this whole blood potency values in the 3 assays that we showed before. Furthermore, we distilled all of this information and calculated the average inhibition of these JAK or TYK2 pathways at various doses for this used in this analysis. Now, let's look at the key results in this slide. The first is this table provides the in vitro whole blood potency values for the various inhibitors used in this analysis. First, focusing on cravacity on the right-hand side, the TYK2 IC50 for cravacity is around 42 nanomolar, indicating potent inhibition of this pathway. And this is as expected based on the mechanism of action. Importantly, the TYK2 IC50 at 42 nanomolar is considerably lower than the JAK1/3 IC50 shown here, which is around 1646 nanomolar. Importantly, the JAK2 IC50 for cravacity was greater than 10 micromolar, indicating no inhibition of this pathway even at this micromolar doses. This indicates at first glance that there is greater selectivity for TYK2 compared with JAK1/3 or JAK2 assays, just based on these whole blood values for cravacity. Now let's focus on the other inhibitors that were evaluated in this analysis. These include tofacitinib, upadacitinib, and baricitinib. And as shown in the table below and highlighted in the red box, these inhibitors demonstrated potent inhibition of the JAK1/3 pathway with IC50 values ranging from 8 to 17 nanomolar. They have some degree of JAK2 inhibition as the IC50 values ranged from 30 to around 200 nanomolar. In these whole blood assays, these agents did not meaningfully inhibit TYK2 as the IC50s ranged from 2 to 5 micromolar range. And when we compared the inhibitory potencies or IC50 values in these values were comparable to ranges of values reported in literature reports. One of them is shown in the slide here. As we all recognize, these whole blood IC50s values by themselves provide limited overview of the selectivity of these molecules. To enable a more holistic comparison, we need to integrate these whole blood potency values with plasma concentrations of these agents that are typically achieved at therapeutic doses. This info, wherein we integrated the whole blood potency values with pharmacokinetic profiles to enable a holistic comparison, is shown in the next few slides. The first plot in this graph is for cravacity. So what this figure provides is the plasma concentration time profile, which is on the x-axis you have time, on the y-axis you have plasma concentrations in nanomolar. So you have the plasma concentration time profile of cravacity at 2 dose levels, 6 milligram once daily and 12 milligram once daily, shown in the blue and in the light gray curves there. These doses were selected as these were evaluated in the phase 2 study in psoriatic arthritis that I just mentioned before, And also the 6 milligram QD dose is being evaluated in multiple phase 3 trials in psoriasis. So on the same graph, in addition to the plasma concentration time profiles, you have 3 horizontal dotted lines. These are whole blood IC50 values or potency values in the various assays. So in the pink dotted line here, you have the TYK2 IC50 for deucravacitinib. In the dark gray dotted line, you have the JAK1/3 IC50, and in the light gray dotted line, you have the JAK2 IC50. It is very clear from this figure that the deucravacitinib plasma concentration at both the 6 and 12 milligram doses are above or close to the TYK2 potency value, or TYK2 IC50 in this case, at least for a period of time. This indicates potent inhibition of this pathway, as we expect based on its mechanism of action. Importantly, the plasma concentrations of deucravacitinib are way below its JAK1/3 or JAK2 potency values. For example, if we look at the peak plasma concentration of deucravacitinib, even at the 12 milligram doses, so the peak plasma concentration at the 12 milligram dose is 8-fold lower than its JAK1/3 IC50 and more than 48-fold lower relative to its JAK2 IC50. This indicates that deucravacitinib plasma concentrations at these therapeutic doses are not adequate to elicit any meaningful inhibition of JAK1, 2, 3 pathways. Now, we did a similar, or we plotted similar graphs for the other agents that we included in this analysis. This figure at the bottom plots the plasma concentration time profiles of upadacitinib at 2 dose levels, 15 milligram once daily and 30 milligram once daily, in relation to its potency, similar plots as what we did for deucravacitinib above. The 15 milligram dose is the approved dose in rheumatoid arthritis, and the 30 milligram dose is evaluated across multiple indications. It can be noted here that the upadacitinib plasma concentrations are above or close to its JAK1/3 or JAK2 potency values, indicating inhibition of these pathways. However, peak plasma concentrations of upadacitinib, even at the higher dose of 30 milligram, is still way below its TYK2 potency value. The value here, if you look at the numbers, it's around 33-fold lower than its TYK2 potency values, indicating that upadacitinib concentrations at these dose levels are not adequate to elicit any meaningful TYK2 inhibition. Now let's look at the same plots, but for the other 2 agents, baricitinib and tofacitinib. And similar plots are shown here. And if you look at all these plots in details, the key results are relatively comparable across these 3 agents, upadacitinib, tofacitinib, and baricitinib. And this indicates that plasma concentration of these 3 agents are above or close to their JAK1/3 or JAK2 potency values, indicating inhibition of these pathways, whereas plasma concentrations of all the 3 agents are way below its TYK2 potency value, indicating no meaningful TYK2 inhibition by these agents at these doses and plasma concentrations. As discussed before, we further integrated these PK values and whole blood to calculate daily average inhibition of each pathway for these agents. So let's look at the distillation of all of this into the daily inhibition value. So this graph demonstrates the daily inhibition of JAK1/3, JAK2, or TYK2 activity for deucravacitinib at the 6 and 12 milligram once daily doses. The average daily inhibition of JAK1/3 activity is shown in a dark gray bar. For JAK2 activity is shown in the light gray bar, and TYK2 activity is shown in the blue bar. It's apparent from this figure that at clinically relevant doses, deucravacitinib inhibited TYK2 activity by more than 50% over a 24-hour period at these 2 doses, which is, as expected, consistent with this mechanism of action. Importantly, deucravacitinib exerted minimal effects, less than 1%, of JAK1/3 or JAK2 activity. This indicates that deucravacitinib is a selective TYK2 inhibitor and does not modulate the JAK1/3 or JAK2 pathways. Let's look at similar plot, but for the other agents included in this analysis. If you look at this plot, if you look at tofacitinib, upadacitinib, or baricitinib, all these three agents exhibited varying degrees of inhibition against JAK1/3. For example, the daily average inhibition ranged from 60 to 94%, and JAK2, which ranged from 23 to 67%. Importantly, there was no meaningful inhibition against TYK2 because the numbers were less than 2% for these agents. So summarizing this poster and this analysis, this confirms that deucravacitinib is a highly selective allosteric TYK2 inhibitor with minimal or no activity against JAK1/3 or JAK2 pathways. Selective TYK2 inhibition is consistent with the reduced potential for treatment-related toxicities like laboratory parameter abnormalities among patients treated with deucravacitinib. Some of these lab parameter changes are effects that are generally associated with JAK1/2/3 inhibitors. Conversely, the JAK1/2/3 inhibitors included in this analysis, like tofacitinib, baricitinib, and upadacitinib, did not exhibit TYK2 inhibition. Hence, the undesirable adverse effects associated with these agents, as noted above, are unlikely to be related to TYK2 inhibition. These results suggest that deucravacitinib is a distinct therapeutic class compared with inhibitors of the closely related intracellular signaling kinases like JAK1, 2, and 3. With that, I would like to thank again the organizers for inviting me to give this talk on behalf of the authoring team. Thank you.
I'm Laure Gossec, a rheumatologist from Paris, France. It's a pleasure to present to you the effect of deucravacitinib on psoriatic arthritis in terms of patient-reported outcomes, analyzing results from a phase 2 study. Now, deucravacitinib is a novel type of drug. It's an oral selective TYK2 inhibitor, which has a different mechanism of action from JAK inhibitors. And in last year's Congress, we showed the results of ACR 20 response at week 16 for both doses of deucravacitinib, either 6 milligram or 12 milligram once daily versus placebo. And we found ACR 20 responses around 53 to 63% versus 32% for placebo. In the present analysis, we are assessing the effect of deucravacitinib versus placebo on quality of life specific to psoriatic arthritis, which is assessed using the PSAID 12 and PSAID 9 impact of disease scores. Patients are described in table 1 of the poster, and there are around 70 patients in each of the 3 treatment groups, placebo and both doses of deucravacitinib. Disease duration is around 5 years, swollen joint count is around 10 to 11, and patients had quite high impact on their quality of life due to their disease, since we can see that the PSAID score was around 6 at baseline, and this is a 0 to 10 score, where 0 is perfect status. The main result of this trial in terms of quality of life is that there were improvements in PSAID 12 and PSAID 9 total scores at week 16, with improvements around 2 to 2.3 points in the deucravacitinib doses versus improvements of 1 point in the placebo dose, indicating that deucravacitinib clearly improves impact of disease in these patients at week 16. Results were very similar for the PSAID 9 and PSAID 12 scores, whereas PSAID 12 actually includes more aspects of quality of life than PSAID 9. When we looked at links between improvement in PSAID scores and achieving response for other patient-reported outcomes, we found that patients who improved in terms of PSAID 12 total scores were also those who were improved in terms of fatigue using the FACIT or in terms of quality of life using SF36. And correlations between improvements in PSAID and other impact scores were quite high, with correlations around 0.6 to 0.8. Therefore, in conclusion, this work showed that deucravacitinib improves impact of disease at both doses more than placebo at week 16, and that patients who improved in terms of PSAID scores also improved around 3 points on a 0 to 10 score for multiple other patient-reported outcomes. Have a lovely Congress.
Hello, my name is Walter Maximovich, and I'm Professor of Medicine at the University of Alberta and Chief Medical Officer of Care Arthritis. And it's my great pleasure to present this work entitled Filgotinib Treatment Results in Reduction of Inflammatory Biomarkers Associated with Disease in Patients with Ankylosing Spondylitis. This work is based on the Tortuga study, and Tortuga was a 12-week randomized, double-blind, placebo-controlled phase 2 study in patients with moderately to severely active ankylosing spondylitis and who had an inadequate response or intolerance to 2 or more non-steroidal anti-inflammatory drugs. And patients were randomized 1-to-1 to receive either filgotinib, 200 milligrams daily, or placebo once daily for 12 weeks. And there were 58 patients in each treatment group. And as you can see from the diagram, biosamples were taken at baseline, week 1, week 4, and at week 12. And this slide illustrates the biomarker changes following treatment with filgotinib. And what you can see is that there are 5 clusters of biomarker response. The clusters being identified based on the kinetics and magnitude of change from baseline with filgotinib treatment. So, for example, the cluster 1 would be those biomarkers with quite a rapid and strong decrease in the level of the biomarker, with a percentage decrease of at least 40% from baseline. And an example of that biomarker would be C1m, which is a metalloproteinase degradation fragment of type 1 collagen. And you can see a very rapid decrease that's already evident at week 1 from baseline. A second cluster would be those biomarkers demonstrating a decrease that is more moderate, with a percentage change of 12 to 40%. And cluster 3 would be those biomarkers with a decrease that is less than 12%. C3m, which is a neoepitope formed by metalloproteinase 9 mediated degradation of type 3 collagen, is one such example. There is a decrease. It's not quite as pronounced as with C1m. And C6m is another biomarker of tissue turnover. Again, there is a decrease, but not quite as pronounced as the decrease that we observe with C1m. And then there are biomarkers that demonstrate a delayed decrease after the institution of treatment. And that example would be metalloproteinase 1. We also have examples in cluster 5 of biomarkers that demonstrate a gradual increase after the institution of treatment. For several biomarkers, a decrease was demonstrated towards the healthy volunteer range following treatment with filgotinib. And on this slide, we see a decrease in levels of ICAM1 and VCAM1 that's approaching levels that we would expect to see in healthy volunteers. The decrease in serum amyloid A protein and tumor necrosis factor does not quite attain levels that we see in healthy volunteers. On this slide, we now see the correlation of the change in biomarkers between baseline and week 12 and change in clinical characteristics, in particular, measures of inflammation. And these include the ASDAS, the C-reactive protein, the spinal pain, the BASDAI, and the spine spark MRI inflammation scores. And these are all changed from baseline to week 12. And we see that the correlations are quite pronounced between the change in the biomarker and the change in the ASDAS and the change in the CRP. There are fewer correlations noted for change in the BASDAI and change in spine spark MRI inflammation scores. And to summarize the changes that we see following the institution of filgotinib treatment, we first of all see changes in markers of inflammation, decreased levels of C-reactive protein, and several other biomarkers that are very well known to be associated with inflammation. Of particular interest, there was no significant impact on biomarkers such as interleukin 17, 21, 22, and the P19 subunit of IL-23. So these are biomarkers that we would typically see in the IL-17, 23 axis of inflammation. We see reduction in biomarkers of angiogenesis as indicated by vasoactive endothelial growth factor. And finally, we see a reduction in biomarkers indicative of matrix remodeling, particularly metalloproteinase 1 and the collagen degradation fragments of type 1, 3, and 6 collagen following metalloproteinase degradation. And these are all reduced after filgotinib treatment. So in summary, we do see significant changes in certain biomarkers, typified by a decrease in interleukin 6 and several matrix remodeling biomarkers. But interestingly, no change in the IL-17 pathway cytokines. We also see reductions in several biomarkers to levels comparable to those observed in healthy volunteers, but not for all biomarkers. We see known associations of several inflammatory biomarkers with C-reactive protein, and this is particularly noted for the matrix remodeling biomarkers, C1m, and the metalloproteinase biomarker, MMP1. We see that filgotinib decreases a wide range of biomarkers implicated in the pathogenesis of ankylosing spondylitis, including those involved in inflammation, tissue remodeling, angiogenesis, and cellular trafficking. Thank you so much for your attention.
Hey, thanks for joining us for this edition of our ACR Highlight Series. I hope you enjoyed these presentations. And don't forget to subscribe to CSF Podcasts on Apple, SoundCloud, or wherever you get your podcasts from so you don't miss out on our Congress content or any of our other usual monthly content. You can also visit the CSF web pages at cytokinesignaling.com, where you can access a whole range of resources, from the monthly slide summaries of latest papers to accredited CME courses and even more content in between. Thanks for listening.